My name is Teresa, and this is Taylor, and our project is on determining whether human disturbance has an effect on soil invertebrates in new and old growth environments. This test was designed to test whether invertebrates from high diversity or low diversity has an effect. The experiment was designed, the diversity is an important role in the biology community because of the roles that species play will boost the ecosystem productivity. Soil invertebrates are a great contribution to soil quality and their roles in soil processes. When further researching on disturbance and diversity, I ran across two articles. The first article, published in the Soil Biology and Biochemistry, Disturbance-Diversity Relationships for Soil Fauna Are Explained by Burt et al. In 2014. Researchers studied disturbance-diversity relations for soil invertebrates in salt marsh or coastal grassland regions that are regularly flooded by seawater. They tested two environments in this area. The first environment was a cattle-grazed region where cows typically ate their food, and the second region was the area that was flooded with seawater. They collected soil invertebrates using the pit trap that allowed the soil invertebrates to fall into 70% ethanol to be preserved until they were taken back to the laboratory. Once they were retrieved, they were classified by their genus or species. The study showed that seawater lands had a negative effect with diversity and showed lower diversity results. In the second article by Burt et al. In 2016 did a study hypothesizing coastal lines that had more human outside activity would have lower levels of diversity. To complete this experiment, researchers compared diversity in plants and small organisms in coastal sand dunes. There were two environments that looked at this experiment. The first one was coastal sand dunes that had high human activity and outside activity. And the other one was a private sand dune that had little human activity. The results were collected through the experiment showed a correlation between disturbance in the plants and the small organisms. Researchers concluded that the areas that had the high human activity showed lower mountain diversity due to the disturbance in the area. Once reading both of these journals, we hypothesized that orders in the old growth region will have a higher diversity rate than orders in the new growth region due to human disturbance. Our study was conducted at the University of Mount Union's Nature Center. We went to two sites. The first site was an old growth site that had little human activity and was out of areas that had walking trails. The second site was a new growth site which had a bar nearby, a walking trail, as well as a pond and more human activity. Leaf litter, or piles of leaves that were picked up from the top all the way down to the soil, were collected and placed into gallon-sized baggies from each site, as well as individual leaves to determine dominant trees in that area. The leaf litter was then taken back to the University of Mount Union's Nature Center laboratory, and the release funnel method was conducted. Leaf litter was placed into separate large funnels that had ethanol attached, as well as wire screening so that leaves cannot fall into the ethanol. The release funnel was then placed under the light for 1 week to allow soil invertebrates to move away from the light and down into the ethanol for preservation. The following week, the ethanol was removed from the release funnel and soil invertebrates were further examined. 3 milliliters at a time of new growth and old growth ethanol was placed into a petri dish and viewed under a dissecting scope to look at the characteristics of invertebrates. This was done until all ethanol was removed from the tubes. The invertebrates were then classified by order using the John Mayer Quick Key to Soil-Dwelling Invertebrates. Once all invertebrates were classified, the Shannon-Weiner indices were calculated for each site. In this experiment, our dependent variable was the soil invertebrates found at each site, and our independent variable was the old and new growth sites.
For the results section of this experiment, Shannon-Weiner indices were calculated for the soil invertebrates found in each environment. The indices are used to determine diversity found in both of the environments. There are 5 columns in these indexes where the first is the order of the soil invertebrates. The second is a N sub P or N sub I column, which is the total number of organisms in each order, and then a P sub I column, which is the relative frequency, and two calculation columns, where the first calculation column is the log of P sub I, and the second is the log of P sub I times P sub I. H prime is calculated through this index because it determines the diversity that is found. H prime is found by adding up all of the values found in the log of P sub I times P sub I column and multiplying by -1. The H max is found by also using these indices by taking the log of the total number of orders found from each of the environments. For the old growth environment, which was an environment that was located in a forested area and had most of the trees being cucumber magnolia, sugar maple, and American beech, the H prime value was 0.772, and the H max value was determined to be 0.845. The two most abundant soil invertebrate orders found in this environment was Mycoptera and Patura. This was also done for the new growth environment, where the new growth environment was located by a pond and a stream, and had the most common trees being red maple, sugar maple, and black cherry. The H prime value for this environment was determined to be 0.754, and an H prime or H max value of 0.954. And the two most common soil invertebrates found in this environment was the Columbella and Acarina. For the discussion, for this experiment, it was hypothesized that the old growth environment would have the most amounts of diversity, and after comparing the diversities between the two environments, it was found that the results did support this hypothesis. Where the H prime value of the old growth environment is 0.772, and the H prime value of the new growth environment was 0.54. The total stability of each diversity were then calculated by comparing H prime to H max, where this helped determine how stable each environment was. When comparing the H prime to H max of the old growth environment, it was found to have the comparison value was 0.073, and the new growth comparison had a value of 0.2, indicating that not only did the old growth environment have more diversity, but it had also a more stable diversity. Some of the differences that can be seen between the two diversities found could be not only the disturbance that the new growth had, but the environment of which the samples were taken, where the new growth environment had a more rich and moisturized soil compared to the old growth, and the different types of trees found in each environment, which could lead to the different types of soil invertebrates found in each environment. When comparing the results of the two previous studies, in the study conducted by Berg et al. In 2014 and the study conducted by Frey et al. In 2016, it was found that in all three cases, disturbances did have a negative impact on the diversities found. And this is also supported in our results, where it can be found that the old growth environment had a higher diversity rate, because it did not experience disturbances where the new growth did. And these are the references.